I guess my next task is to introduce myself and to talk to you about radioisotopes. I'm an internist slash nuclear physician who's practiced in London for about 35 years and have had a long interest in radionuclide therapies. And when I was asked to present this talk, I was asked to make it at a sort of a university undergraduate level so that people who feel that there's a mystery about radioisotopes would become more comfortable with the term. Radioisotopes have been all over the news for the last few months and will likely remain there. I guess I need a tutorial. Pardon? There we are. They've been all over the news the last few months and will remain there for a while longer because the reactor leakage that shut down the reactor at Chalk River has highlighted a worldwide problem with the future of radioisotope supply. And in every country where people practice nuclear medicine, this is an issue. The price of isotopes has gone up between 200% and 300% in the last 3 months. And I was speaking yesterday with a physician from Libya who said, well, we haven't had any isotopes in the last 3 months. And I know that's true of other developing countries who at the moment perhaps had small nuclear facilities that they can't afford to operate anymore under the new price structures. First thing that I'd like to draw your attention to in relation to isotopes is just to think about the structure of the atom. And there is, in every atom, as you know, a nucleus that has its mass is made up of a number of protons and neutrons, and there are electrons that are sort of skittering around the outside. It's not quite like that, but the quantum mechanical model doesn't allow you to draw a picture, so that's how we'll leave it. Neutrons are particles that don't have any charge. They're neutral, and they are the product that are the form of radiation particle that's used to bombard targets to prepare isotopes in a reactor. Now, what I want you to just be reminded of, you probably saw this in high school or in university chemistry. This is the periodic table of the elements, and this is the shorthand for all the elements that exist. The heaviest element that exists in nature is uranium, 92, and most of the others exist. Technetium, the element that we use in nuclear medicine, actually does not exist in nature and has to be made, and we irradiate uranium in a reactor to make molybdenum fragments, which we capture and distribute, and these decay to technetium. And technetium has very good qualities as an element that we can label to many compounds to image the functions, because it is the functions rather than the structure of the body that are imaged with nuclear medicine. Now, think of this table of the elements as the sort of front of a cabinet, and then we can we can ask the question about isotopes going on from there. The term isotope comes from the isotopos, meaning in the same place, and so the isotopes of each element are in together with all of the stable elements, and they vary in terms of the number of neutrons. Now, if you consider that each one of those elements was a drawer in a filing cabinet and you open it up, for each element you see there are dozens of isotopes. And up here, I can't make it out from here, but there's iodine. There are about 45 isotopes of iodine, of which 3 or 4 are useful in medicine because of their radiation properties and their half-life, and we use them for different procedures. So people ask us, what is radioactivity? Because it's kind of a scary thing that many people with political agendas have used to frighten us into thinking certain things. Think of a radioactive isotope as one that has been induced to have an unstable ratio of protons and neutrons, and at some point that instability has to be resolved, and so it's like a rock sitting on the top of a mountain that's just precariously caught and held up there. Along comes a heavy rain and washes away the support structure, and the rock goes to the bottom at some random time. It releases a lot of energy and assumes a stable position at the bottom. And that's what really, it's a good model for radioactive decay. Now, how do we make radioactive isotopes? Well, in nature, we have all kinds of them, and they came originally from supernovae when stars blew up in the past, and in the last day of the 5 or 6 billion years of a star's life, all the elements heavier than iron are made in a few hours, and then the star blows up in the spectacular explosions. And these elements, these isotopes that I've listed here, are radioactive isotopes that are in our environment that have a long half-life, except for carbon-14. Now, it's got a different story, but they have a long half-life, and so these isotopes were made when the star exploded, from which the rubble eventually compacted to form our Earth, and we still measure these in our environment. For instance, these days, every shipment of bananas that comes into the United States sets off the radiation alarms at the border because of the natural concentration of potassium-40 in bananas. If the Americans imported oranges, which I think they do not, they would have the same problem there. So there is a significant amount of natural radioactivity in our environment, and there is both of the isotopes of uranium that exist on Earth are radioactive with long half-lives. And those are isotopes that are critical to the functioning of reactors through which we make isotopes. Now, nuclear reactors irradiate the targets that we select with neutrons. Cyclotrons are another kind of radiation machine that you may have heard of. They are machinery that uses electricity to power up, and they can irradiate targets with charged particles. So if we want to make technetium, we would irradiate, or iodine, or yttrium, or lutetium, we would irradiate a target with neutrons. If we want to make fluorine for a PET scan, and fluorine is a neutron-poor agent, we would irradiate a target with charged particles from a cyclotron to make a neutron-poor product. So we have this range of products that we can make. And so here is, if you like, the blue in the center here is in each square represents one of the stable isotopes in our Earth. The red ones to the right are produced in reactors. The ones to the left are produced by neutrons. So, and there are, as you can see, not just the 92 elements here, but there are dozens of isotopes of many of the elements, so that we have several thousand potentially useful isotopes. And let me show you what a difference a neutron makes. Just think about fluorine-19. That's the stuff that's in our drinking water and in our toothpaste to prevent tooth decay. One neutron less, and it is fluorine-18, and we will use it for an FDG PET scan. Think of carbon. Here's carbon-12, and that's the stuff that made up about, after you eliminate water, about 40% of your breakfast this morning. Carbon-13 is another stable isotope that's of interest to archaeologists, because when they dig up ancient midden heaps from 20,000 years ago, and they ask the question, what did the people eat 20,000 years ago, the enrichment of the midden heap with carbon-13 tells you whether that they were eating a lot of seafood. Carbon-14 is produced by solar radiation in our environment, and it's used for carbon dating. So if you find something that might be somewhere in the order of 5,000 to 20,000 years old and you want to date it, carbon-14 is a clock that we can use to date things. Over here, carbon-11, which is a short-lived isotope that is possibly going to have a significant future in medicine as a PET scanning agent, and Dr. Oberg's group, for instance, use carbon-11 ephedrine for PET scans. So a neutron, more or less, makes a huge difference, and you can appreciate what a powerful toolbox we have here in the isotopes. Well, just having the isotope is great, but we need to do other things to craft it and to give it some smarts. And so typically we want to incorporate the isotope with a chemical to make a radiopharmaceutical that will localize in the target tissue. And so we require that there be incorporation of the isotope into a compound. Sometimes, as in thyroid cancer, which is my particular area, we use the elemental iodine. And during the time when we can't get technetium for bone scans, we can use fluorine-18, and that's being done in Quebec just now for bone scanning. But usually we have to make a compound, like we have to add I-131 to MIBG to get the right kind of localization in neuroendocrine tumors. And we might, these incorporations make for radically different molecules with radically different uses. Fluorine-18 for bone scanning, potentially fluorine-FDG for imaging many kinds of tumors that have an active or activation of glucose transporters. Doesn't work as all that well for neuroendocrine tumors unless they're quite dedifferentiated. People are evaluating fluorine-18 thymidine as a way of imaging cell division in tumors and assessing the response of tumors to treatment. And fluorine-18 fluorodopa is another agent that's been used in imaging neuroendocrine tumors, and I know that our colleagues in McMaster University are keen to do some work with that agent. Now, when we make a radiopharmaceutical, we can't anymore just say, gee, we're very clever, we've got a great radiopharmacist who's made this compound, it chromatographs properly, we'll inject it into a patient. I've been around long enough, we used to be able to do that, but nowadays you have to go to Health Canada and you have to demonstrate the proof of what you've got and the efficacy in order to, and you have to demonstrate that you have the kind of facilities in which you can do safe manufacturing, and so it takes a long time to secure approvals for new agents. So we come to the machinery of isotope production. Cyclotrons I mentioned. The first cyclotron was built in the 1930s in Berkeley by Ernest Lawrence, and it took so much energy that he could only operate it at night. And even at that, people said that when they turned on the cyclotron in the evening, you could, if you were standing outside, you could see the lights of the city dim. So there are issues about the operation. But we can bombard targets to make a number of isotopes that are of importance to medicine and to neuroendocrine tumors in particular. Here's indium-111, which is used and has already been mentioned as an imaging isotope. Gallium-67, not so much anymore. Thallium, not so important for neuroendocrine tumors. I-123, if you can get it on the right sort of timing, is great for labeling MIBG if the issue is diagnosis rather than therapy. And we're kind of excited about the possibilities of gallium-68 generators because you don't need a cyclotron in your facility to do that. You can buy a germanium source, and like moly technetium, you can milk the gallium from the germanium, and this is a good PET scanning agent. And there are some nice papers from Europe about this. I've spent about 5 years trying to get approvals for a gallium-68 generator that we could use in humans, but we have not been successful so far. Now, nuclear reactors are very different machines. The first one was built in 1942 in the University of Chicago underneath the bleachers at the football stadium, and it was built in order to prove in principle that uranium was fissionable and that there was some potential to build a weapon of war with this. Along the way, we've found some redeeming uses for nuclear technology, and so these days we bombard neutrons to make radioisotopes, and one of the products we have is we call fission products. And this is the situation at Chalk River. When the reactor is operating, we actually get a pencil of enriched uranium-235, which is the dangerous bomb-grade isotope, about 90% pure. And because this product is brought on that site, the security is unbelievably tight and is dictated by the Americans. And this product, these uranium pencils are irradiated in the reactor for about a week. And the uranium typically breaks not quite in the middle to form two fragments. And one of the common fragments is molybdenum-99. Other common fragments are I-131, strontium-90, and yttrium-90, which are also medically important isotopes and obtained from that source. They then have to, after about a week of irradiation, take this pencil of uranium out of the reactor and under very heavily shielded remote conditions dissolve it in acid and extract the molybdenum and look after very, very radioactive liquid waste. So it's a complex process, and we hope that we'll get that going again next year. The issue around the crisis is that there are only 5 reactors in the world that are equipped to make isotopes. You can't just do this with any reactor. And the 5 reactors are all research reactors, the youngest of which is 46 years old. NRU is the oldest at 52. And the world has been coasting with a senescent technology. And people like you and I are obviously very concerned as to what the future will be. We'll get the holes patched in NRU next year. It's going to cost $80 million, they say. And the reactor may be allowed to operate for a few more years. But there has to be a solution coming forward before that breaks down finally and decisively. In addition to irradiating uranium itself and extracting products from it, we can also put in other targets and irradiate specific targets to get specific isotopes like, well, that's wrong, that's lutetium-177, germanium, as I mentioned, cobalt-60, and there's a household isotope that we're all interested in. It's americium-241, which is the product of irradiating plutonium. And this is the thing that drives the smoke alarm in your house. And without americium-241, we would not have smoke detector technology. So let's understand this, that none of the technology that we have and that we are currently critically relying on to develop radioisotopes was initially designed to produce isotopes. It's all, in fact, in the U.S. National Regulatory Commission, the official word for isotopes, medical isotopes, is byproducts. So, you know, they didn't have this in focus at all, and it's popped out. But without that, we would never have developed the field. We have to develop a sustainable and robust replacement technology, and there's a lot of scrambling going on behind the news reports these days to try and reach toward a conclusion as to how we do this for the long term. Medicine is, yesterday I spent 12 hours at the Bruce Nuclear Station with 20 of our students from the University of Western Ontario, and the point was it was like a family reunion where the nuclear medicine people met the nuclear engineers, and they're like the long-lost cousins. And we recognize that nuclear is one industry and that we don't have the resources to build the facilities that you need for isotopes and for nuclear engineering and for basic physics, and we all have to get together and build shared facilities to make these things work. So there's one industry, we're all part of it. If you show up in an emerge department these days needing repairs of some kind or other, the sterile materials that are brought to your trolley side to help you have at least a 70% chance of having been sterilized by radiation. That's another application. And about 50% chance that that cobalt that was used to irradiate these supplies to sterility was produced at the Bruce Nuclear Station. And it's one of their major byproducts of operating that reactor. So we have we have to approach this as an integrated issue that has to serve aspects of science as widely diverse as I've indicated, from anthropology to geology and physics and medicine and nuclear power. Now, I've said a number of the things here already. Of the 5 reactors that are operating, the next oldest one is due to go down for an extended shutdown next year. That's the one in Petton. So if it goes down before we get NRU back up again, and there may or may not be choice about that, then we are really going to have a problem in the world, because of the 5 reactors, 4 of them were essentially running at capacity when the NRU reactor broke down. And NRU was producing more than 50%, and it's the only reactor that had additional reserve capacity available. The U.S. NRC has looked at requirements, and they estimate, as I say here, that the requirements for isotopes will increase about 12% per year for at least the next 3 years. And the Government of Canada, just before we had the NRU breakdown, decided, without consultation and without asking us about any of the consequences, to end the Maple reactor project. And so we are losing a huge Canadian business. It's a bit like the Avro Aero story all over again, but I won't belabor that. So isotopes in medicine support functional imaging. The uses in therapy are increasing continually. If you look at the Journal of Nuclear Medicine, which is our major publication, and it is ranked in the citation index with 80 general medical imaging journals, the Journal of Nuclear Medicine is now #1 in the imaging journal citation index, which means that people are taking more and more notice of what nuclear is able to do for medicine. And that's because the sensitivity of radioactive isotopes is so much greater than that of morphological imaging like CT or MR. And the sensitivity of PET is about 1 million times greater than the sensitivity of magnetic resonance spectroscopy, which would be only the only roughly competing technology. So we are going to be needing isotopes into the indefinite future. For you people who are interested in neuroendocrine tumors, you can see there are quite a number of isotopes that we're interested in, and we need combination of reactors and cyclotrons. When the NRU reactor is operating, they were making lutetium, and I think in some cases those of you who have gone to Europe for therapy have there met Canadian-produced lutetium, and you had to go there to join up with it, which is an embarrassment and a shame, I think. And of course, we have some PET isotopes that are also of importance. So why would we add radioactivity to drugs? We've heard about the importance of somatostatin. If somatostatin works, what does radioactive therapy do better? And we have to be sure that that's the case, that it is better, before we talk about it or deploy it. We can confer cytotoxicity on drugs that are otherwise not toxic. So if we want to kill cells, we get what are called crossfire effects. That is, if you have cells that are heterogeneous, some of them have the receptors that you're targeting and some of them do not, there is a possibility of irradiating the cells that are not actually taking up the tracer through the crossfire effects and the reach of the beta radiation from the site of the breakdown. And so that we can accomplish a better cell kill, we hope. And I think that's true. And there's also a recently revealed additional effect that we call the bystander effect. And the effect is this, that at least in the lab, if you can irradiate a single cell and know which cell it is, and kill it, the nearby cells that are not irradiated but are touching will get the message that it's time to die. And this is a chemically mediated kind of message, and so by irradiating one cell, one is able to kill quite a number of cells. Just how this would operate in a tumor is not, or to what extent it does, is not yet clear, but it's a very encouraging observation. And the reason we need more than one radioisotope for therapy is that the path lengths of the beta radiation have to be a considerable portion of the tumor diameter in order to reach into all the places. There are what we would call sanctuaries. There might be a part of the tumor that does not have enough blood flow to pick up, isn't presented with as much of the radiotracer. There are hypoxic zones, perhaps, and there are regions where the tumor may have mutated and no longer have the receptors we want. And so we need all of the tracers that are available in the international literature in order to program the best treatment for an individual patient. Here's an example of clonal heterogeneity. Here's a patient with a carcinoid, and we have an I131 MIBG scan and the same slice through the liver of an octreotide scan. And you can see MIBG targeted a lot of things here, but notably did not target this large metastasis in the left lobe, whereas the octreotide has dominantly targeted this site. And this tells us one radiopharmaceutical by itself isn't going to be enough, and that we have to have complex and additional protocols available to reach out to destroy the whole population. Octreotide imaging could be improved. Here are some, if you look at the binding coefficient of these products to the somatostatin receptor, that's the native product, and the lower the number here, the stronger the binding. You can see octreotide does not have nearly the same kind of binding affinity as the native protein. The problem is that the native somatostatin has a half-life in the blood of less than a minute, whereas this is fairly stable and hangs around for hours, and there's time to take it up. Some of these other agents, the gallium and indium labeled agents, again have binding affinities that are near that of somatostatin. And we don't have these available in Canada yet, and I guess that's because we don't have an industrial concern that's really pushing to help us obtain them. Now, the puzzle that I'm presenting you with has 4 parts, and I'm going to try and just indicate a solution, and hopefully we'll have a chance to talk about it later in the meeting. I think there are 4 issues around at least the isotope portion of the neuroendocrine tumor dilemma. There's the issue of isotope availability, which we've discussed. There's the issue of radiopharmaceutical approvals. And Health Canada has said to us, well, ask us, we'll approve stuff. And the real problem we have is that approvals are a federal issue, reimbursement is a provincial issue, and when we go to the province of Ontario, to OHIP, and ask for, through the OMA, for reimbursement for the activities that are necessary to deal with neuroendocrine tumors, they say, show us the phase 3 evidence. I was involved for at least the past 5 years, I've lost track, trying to obtain a formal agreement to reimburse the cost of octreotide scans. And we do them through a bureaucratic wrinkle that I won't discuss, but we don't actually have approval in Ontario to perform and to be reimbursed for octreotide scans, let alone all the other things we would like to do. And the other thing we need is, to make this work, is to adopt a culture of continuous learning amongst ourselves and to integrate ourselves together as teams, as Dr. Gospodarowicz and her colleague have already spoken. So we have the crisis at Chalk River. There is, I should tell you that even therapeutic MIBG, which I gave the first therapy with MIBG in Ontario in 1987, and it's still not an approved drug for that application, and we have to go through the compassionate program of Health Canada each time. There's a huge lag in Ontario around the approval of PET radiopharmaceuticals, And that's, again, another story that we've spent about 10 years on trying to fix. And again, the Ministry of Health says, give us your phase 3 evidence before we'll approve these. And only this last month did they agree to fund for any kind of PET scans. And we need lutetium, yttrium. And so that's... So I think what we need to do, these are all orphan drugs. The potential market will never reimburse for the cost of the trials that are being requested or for the cost of the approvals process, because a typical pharmaceutical company will look at a drug and they'd say, well, worldwide we'd need about $1 billion in sales to make it fly. Well, for all radiopharmaceuticals in the world taken together, the market is less than $2 billion, let alone what it would be for, one Octreotide product needing approvals. So our dilemma is that the approvals process is weighted against us. And so what we think is needed is that we should develop internationally compatible registries and go forward in a sort of collaborative center of excellence style without regard to geographical priorities or whatever, but wherever things can be done best, and to collect evidence in a registry through the practice of protocols that we might all agree on in the main. So that's something we would very much like to see going forward. The hospitals won't let us order these things, bring them in-house until there is reimbursement. And we have to have a vision that transcends all the local fiefdoms and geographic domains. Even the provinces are in some cases too small because of the population bases of a number of our Canadian provinces are such that they probably don't even notice that they have an issue about care provided to neuroendocrine patients. And we've been told, for instance, that although we were doing a lot of neuroendocrine therapies, that we can't take patients from outside of our LIN anymore, even though the out-of-province patients were paying. So there are issues about developing, I think, a national or even an international vision to advance patient care in this area. Continuous learning, we just need to have academically oriented specialist teams. We need to have address a sufficiently large population to keep teams busy, international professional connectivity, industry collaboration, and tools for evaluating outcomes. And where our goals should be that we should all, patients and physicians, find some interconnected satisfaction in so doing. So my message is consistent with what you've already heard. We can't do it alone. The health system left to itself won't develop a vision for the provision of these services. And patients and their advocates are very important to help us articulate the needs and promote to the profession and governments that these needs should be addressed, because you're all citizens. And I hope we'll have an opportunity to talk about that later in the session.